Hi, class of 2024. So this is your first class, biology of periodontal tissues. So we will be focusing on the gingival features, mainly what is gingiva, what are the different parts of gingiva, and what are its functions. And then we will be checking about the defense features of mechanisms which the gingiva plays in maintaining a very good oral health in our oral cavity. So we'll go about a little about the introduction about periodontology, and then we'll move about telling about more about the gingiva. Thank you. Textbooks recommended for the subject of periodontology would include Clinical Periodontology by Newman and Carranza, the 13th edition. You can also follow the 10th edition for your reference. Clinical Periodontology and Implant Dentistry by Jane Linde, the 6th edition. So this comes in 2 volumes. The first volume consists of more of the basic of periodontology. The second volume consists of more of the clinical aspects of periodontology. So to start with, what is periodontology? Periodontology is a scientific study of periodontium in health and disease. So we will define what is periodontium in the later aspect of this slide. Whereas periodontics is the clinical specialty of dentistry that is concerned with the prevention, diagnosis and treatment of functional and structural diseases of the supporting tissues of teeth or their implant substitutes. So as I told you, periodontium, the supporting tissues of periodontium include gingiva, alveolar bone, cementum, and periodontal ligament. So all this together constitute your periodontium. So here you can see a healthy gingiva of a 17-year-old girl. So you can very clearly find out that the gingiva is a little coral pink color in this individual with good attached gingiva, good amount of attached gingiva, mucogingival line, the alveolar mucosa and the interdental papilla. So we will find out what are the other inner aspects of gingiva in the later slides. So from this aspect, from this lesson, we would like you to know what are the 4 normal structures of periodontium, recall at least 6 clinical characteristics of the normal gingiva, and discuss the relation between microscopic and macroscopic features, describe the various age changes in periodontal structures. Periodontium. The tissues that invest and support the teeth, namely gingiva, periodontal ligament, alveolar bone and cementum. So these 4 constitutes your periodontium. So here you can see a diagrammatic view of the gingiva, wherein the area between your sulcus and your gingiva, that is this area within your sulcus area, is called as the crevicular epithelium or the sulcular epithelium, whereas the outer gingiva is known as your oral enamel epithelium. Apart from that, the area which is attaching the whole gingival structure, that is the epithelial structure, to the tooth is your junction epithelium. So epithelium is of 3 types when told about gingiva. Apart from this, you do have your periodontal ligament, wherein there is a support of the fibers are present which helps in attachment of the tooth and the gingival structures, and then you have your cementum, and then you have your alveolar bone. So in total, the gingiva, periodontal ligament, alveolar bone and cementum constitutes your periodontium. So again, to tell about this in radiographically, so here you have your picture wherein you do have your bone, alveolar bone here, which can be seen with the help of your radiographs, and a thin lining of periodontal ligament space surrounding the tooth, which is a radiolucent line, which you can see, a black radiolucent line. Up a little away from this, you can see one more white line which depicts your radiographic lamina dura, or otherwise your alveolar bone starting, so or your cribriform plate. So this is a normal picture of your periodontium, and your bone generally starts at around 1 to 2 mm from the CEJ. So you can see the tooth which is there in situ with all its structures present. So again, to start with, you can also tell about your periodontium, wherein you have your alveolar bone followed by your periodontal ligament. Cementum is that attached with the tooth surface, and then the gingiva which is there on the outer circle. So all this together is a periodontium, and periodontology is a scientific study of periodontium both in health and disease. So to start with the first structure, that is your gingiva. The gingiva is a part of the oral mucosa that covers the alveolar process of the jaws and surrounds the necks of the teeth in this fashion. So you can very well see how your gingiva is placed. So this is a little coral pink in color and a little scalloped area around each of the tooth surface which surrounds the necks of the teeth. So there are three main parts in your gingiva. The first part being your free gingiva, the second part being your attached gingiva, and the third part being your interdental gingiva. So free gingiva constitutes of the area just above your teeth. So this area lodges your sulcus, and this is called as the free gingival area. It is not attached to the tooth or the bone within, so it is called as free gingiva. The second part constitutes your attached gingiva, that is the area beneath your free gingiva to the alveolar mucosa. This belongs to, called as attached gingiva because it is being attached to your underlying bone. The third one is your interdental papilla, that is areas between the teeth. This small triangular area is called as your interdental gingiva. So what is free gingiva? So we will speak about this. Free gingiva is the area from the gingival margin to the free gingival groove at the level of the CEJ. So this area is called as the free gingival margin. This can be separated by means of a probe, and this depth of the gingival sulcus is termed to be 0 to 3 mm. The ideal depth is 0 mm. The clinical depth is 2 to 3 mm. And you do have a histological depth ranging from 0 to at least 1.8 mm. So here you can see that the free or the marginal gingiva is the distance between your gingival margin on the top till the free marginal groove. So coming on to the next part, that is your attached gingiva. The attached gingiva is a part continuous with that of your free gingiva. It is firm, resilient, and it is tightly bound to the underlying periosteum of the alveolar bone. This extends from the free gingival groove to the mucogingival junction. In other words, we can tell that the free gingiva and the attached gingiva are separated by the marginal groove, and the attached gingiva and the alveolar mucosa are separated by the mucogingival junction. So coming on to attached gingiva. So as I told you, the area between your attached gingiva and your alveolar mucosa is separated by this mucogingival line, so which you can see very well on the tooth surface. This is generally on the labial or the labial aspects of the teeth, but on the palatal aspects, that is on the upper teeth, the whole palate or the whole mucosa is keratinized, and there is as such no mucogingival junction as seen in the labial sides. Now coming on to the width of attached gingiva, this is an important clinical parameter. So this is the distance between the mucogingival junction and the projection on the external surface of the bottom of the gingival sulcus on a periodontal pocket. So it has been found that on the incisor surface, according to studies, it has been found to be 3.5 to 4.5 mm in maxilla and 3.3 to 3.9 mm in the mandible. And the least width of is found in the first premolar region. In the maxilla, it has been found to be 1.9 mm, and in the mandible, it is found to be 1.8 mm. Now, coming on to what is keratinized versus attached gingiva. So, this is always a confusion telling keratinized gingiva is same as the attached gingiva, but that is not so. So, this is the attached gingiva, that is the area from the free gingival groove to the area from the free gingival groove to the mucogingival junction is the attached gingiva. On the other hand, the keratinized gingiva includes this whole area, that is whichever part is keratinized from the uppermost point on the free gingiva to the mucogingival junction. So this entire area is said to be a keratinized gingiva, whereas attached gingiva is the only part which is attached to the tooth to the bone or the underlying tooth surface. So as I can told you, this is your keratinized gingiva. Now coming on to the interdental gingiva. So this can be either our interdental gingiva, or we also call it as interdental papilla. So this could be either a pyramidal shape or a caul shape effect. Pyramidal shape is usually seen in your anteriors, This is because there's a tight contact between your teeth. So this is pyramidal shape, as you could see. This is mostly seen in the anteriors, whereas in the posteriors you do have two pyramids like this, which is attached by means of a sulcus like this. So it is a kind of a tent shaped, or otherwise known as a caul shaped effect in case of your posterior teeth. So this is present between your buccal and lingual papillae. So it's been found that caul is more susceptible to more disease prone area, because this small area tries to lodge food substances here, and therefore the activity of disease is more in the interdental aspects compared to the facial or the lingual aspects. And therefore when you're going to check for any disease aspect, try to check more on the interdental sides of the teeth. So then coming on to the clinical features of normal or the healthy gingiva. So this, I think I have already taken you a short class, or we had a short Google Meet discussion on the clinical features of normal or healthy gingiva. And I'll be giving you the notes too. So to tell in very short aspects about what are the clinical features. So generally your gingiva is little coral pink in color, because you might have physiological or racial pigmentation. The contour is little scalloped with knife edged margin. So this is called as you have scalloping effect, that is a kind of U-shaped effect, inverted U-shaped effect on the teeth with a knife edged margin. Surface texture is stipple, which means that there is a little orange peel appearance on the tooth surface. The consistency is firm and resilient, and the position of the normal gingiva is at the CEJ or a little above the CEJ. There is no bleeding on probing present, and the size is normal without any gingival enlargement. So now we're coming on the general aspect of gingival epithelial biology. So first was thought to provide, that the gingival epithelium was thought to provide only a physical barrier to infection and the underlying gingival attachment. But now it has been shown that epithelial cells play an active role in the innate host defense by responding to the bacteria in signaling further host reactions and in integrating innate and acquired immune responses. For example, this is by increased proliferation, alteration of cell signaling events, changes in differentiation and cell death, and ultimately alteration of the tissue homeostasis. So what are the functions of gingival epithelium? So there has been a number of functions. So starting with your mechanical, chemical functions, it does provide water as well as microbial barrier. There are signaling functions which are present along with this. Architectural integrity is brought about by the cell attachments, basal lamina, and the keratin cytoskeleton. There are many cells which are present, such as your keratinocyte, Langerhans cells, Merkel cells, and the melanocytes. Gingival epithelium always helps in constant renewal, that is replacement of the damaged cells. So there are also cell-to-cell attachments with desmosomes, adherence, tight junctions, and gap junctions. The basal lamina of the epithelium consists of connecting structures called as the hemidesmosomes. So here you can see the three types of gingival epithelium. So as I told you, the area which is within the tooth structure. So the entire epithelium is connected to the tooth by means of the junctional epithelium, that is your C, and the area which is covering the sulcus area is known as your gingival or the sulcular epithelium. So here you can see the outermost layer having the cells, and the outermost area is your keratinized area having a layer of keratin here. So this is your outer enamel epithelium or outer oral epithelium in the gingiva. Now coming on to sulcular epithelium. So you can see very well in this histopathological slide, wherein the area bounded by your sulcus is called as your sulcular epithelium. The area where the gingiva touch the tooth surface is your junctional epithelium, and the outer area is known as your outer enamel epithelium. So this is sulcular epithelium is a thin non-keratinized stratified squamous epithelium without any retapex, and this extends from the coronal limit of the junctional epithelium to the crust of the alveolar gingival margin. It may act as a semi-permeable membrane through which injurious bacterial products pass into the gingiva and through which the tissue fluid from the gingiva seeps into the sulcus. So this forms, since it is a semi-permeable membrane, we have to be very careful that it is an easy entry for the microorganisms into the gingiva. Now coming on to the junctional epithelium. Junctional epithelium is a stratified squamous non-keratinizing epithelium which is 3 to 4 layers thick in life, but with age it goes to 10 to 20 layers thick. The length of the junctional epithelium is around 0.25 to 1.35 mm. Neutrophils are routinely found in junctional epithelium because they are the first line of defenses, and they are found in this area. This is more permeable than sulcular epithelium. So you can understand that if there is going to be some minimal amount of food deposits also at the cervical level, there is a easy permeability of the food or the microorganisms through the junctional epithelium or sulcular epithelium to the underlying surfaces. So attachment of junctional epithelium to the tooth is reinforced by the gingival fibers. So for this reason, both the junctional epithelium and the gingival fibers are together considered to be a functional unit known as a dentogingival unit. Their functions mainly include giving an epithelial barrier against the plaque bacteria. It allows the ease of access of gingival fluid, inflammatory cells, and components of immunologic host defense to the gingival margin. Junctional epithelium exhibit rapid turnover, which contributes to host parasite equilibrium and rapid repair of the damaged tissue. So here you can see the dentogingival or the gingival fibers which are present. So these fibers are the main ones which constitutes your gingiva or how it is attached to the tooth surface. So here you can see that from this diagram there are gingival fibers or the gingivo-dental fibers are arranged in 3 patterns. So this 1, 2 and 3 are the 3 patterns which are arranged. So fibers arise from the cementum to the crust of the alveolar gingiva, or it could go from the cementum to the outer surface, or it also could go from the cementum to the external surface of your periosteum or the labial plate. And apart from this, you do have small circular fibers like this, and apart from this you also have your transeptal fibers where it attaches from the cementum of one tooth to the cementum of the other tooth. So these transeptal fibers are fibers of both the gingiva as well as your periodontal ligament. So to reiterate again, there are going to be 3 main fibers which are present. One is your gingivo-dental fibers, which are of 3 directions, which you could see here. Second is your circular fibers, and third is your transeptal fibers. So what are the functions of gingival fibers? It is to brace the marginal gingiva firmly against the tooth and to provide the rigidity necessary to withstand the forces of mastication without being deflected away from the tooth surface. It is to unite the free marginal gingiva with the cementum of the root and the adjacent attached gingiva. So there are a number of cells which are present in the gingiva. Fibroblasts, main cell for the production and the destruction of collagen. Macrophages, mast cells, osteoblasts, cementoblasts, osteoclasts, odontoclasts, polymorphonuclear leukocytes, lymphocytes, plasma cells, and undifferentiated ectomesenchymal cells. So coming on to the blood supply of periodontal tissues. So generally it starts from the apical aspect of the periodontal tissues. The arterial branches cross through the periodontal ligament through the alveolar bone and along the through the alveolar bone, and third one is through the surface of the periodontium. So to reiterate again, one is through the arterial branches which goes through the periodontal ligament, and then the second one is through the alveolar bone. The third one is along the surface of the periodontium. The terminal branches of each of these 3 arterial sources form numerous anastomoses and capillary loops like this. Okay, next coming on to the structures of the periodontal ligament. So, the major periodontal ligament fibers and the gingival fiber groups. So, here you can see that in case of periodontal ligament, you do have 5 different types of periodontal ligament fibers, starting with your alveolar crustal group, and then your horizontal group of fibers, your oblique group of fibers, and then your apical group of fibers, and then your interdental group of fibers. Apart from this, you also have your transeptal fibers, which forms a part of your periodontal ligament fibers. Functions of attached gingiva: it gives support to the marginal gingiva, helps to withstand and the functional stresses of mastication and tooth brushing, provides attachment or a solid base for the movable alveolar mucosa for the action of cheeks, lips, and tongues. So, this we will go with the defense mechanisms of gingiva. From this, we would like you to know the various defense mechanisms of gingiva, describe the functions and clinical significance of GCF, that is gingival crevicular fluid, discuss the methods of collection of gingival crevicular fluid and the functions of saliva. So, gingiva prepares and protects itself from various pathogens through the fine tuning of hemostasis. If this balance is lost, then the activity of gingival tissue becomes abnormal, and this change is perceived as periodontal disease. So, defense mechanism is of two types, either the non-specific or the specific defense mechanism. So, non-specific defense mechanism is our innate response, or the innate immune system which we possess. So that is the one which is going to fight against the host, that is the host microbial symbiosis. So your innate mechanism, innate immune response fights against the host, whereas you have the other gingival features which are present, which helps in defending against the bacteria or the viral action. So this is in collectively called as a tissue resistance. So tissue resistance is of four types, that is your anatomical factors, your mucous barrier, which includes presence of saliva, and gingival crevicular, which will again fight against any problems in the oral cavity, epithelial barrier, local inflammatory response. Start with the anatomical barrier. Anatomy of the gingival tissues is designed for effective mastication and clearance of food debris. Any poor functional tissue relations lead to plaque accumulation. Stippling is a functional adaptation to provide resistance to mechanical trauma. Attached gingiva is contoured to allow for the proper deflection of food and is critical for the maintenance of gingival health. Gingival fibers also embrace the marginal gingiva firmly against the tooth, thereby giving necessary rigidity to combat the forces of mastication, and tends to throw away any foreign material into the sulcus. This expulsion is aided by the movement of the teeth and the gingival tissues during the function of the dentition and the pulse beats. Next, coming on to the mucous barrier, which is we will be discussing about saliva and GCF here. So, mucous barrier, saliva, as you know, it is a fluid secreted by salivary glands that begins the digestion of the food. So, you have a tabular column here which describes about the function of the saliva. I think this you would have read it probably from your school days itself, that there is function of lubrication, physical protection, etc. So here we are going to learn a little more in detail. So lubrication is brought about by the products like glycoprotein and mucoids. The probable mechanism is a coating similar to that of the gastric mucin, and physical protection is by means of glycoproteins and mucoids. Cleansing is by means of the physical flow of the saliva. Buffering is by means of bicarbonate and the phosphates present, and the tooth integrity and maintenance is by means of the minerals which are present, such as phosphate and other minerals. Antimicrobial action is by the presence of immunoglobulins, lysosome, lactoperoxidase. So glycoproteins and mucoids in the saliva coat the gingival surface, thereby providing lubrication and physical protection. This flow cleanses the gingival surface by cleaning the debris and the bacteria. The bicarbonates and the phosphates, they act as antacids, neutralizing the acidic bacterial products. Coagulation factors such as 8, 9, 10 plasma thromboplastin antecedent Hedgeman's factor in saliva speed up the coagulation and protects wounds from bacterial invasion. Host defense molecules found in saliva, including the host's innate as well as acquired immune responses, such as antimicrobial peptides, chemokines, cytokines, have a major role in determining the microbial balance and the bacterial colonization. So, you would be a new term here is an antimicrobial peptide. So, these are the early responders of innate immunity that search and destroy the invading pathogens, and they are of many types as shown below, such as cationic peptides, adhesion proteins, proline rich proteins, metal ion chelators, protease inhibitors, and proteins acting against the cell wall. So coming on to gingival crevicular fluid, the second component of the mucous barrier, gingival crevicular fluid is regarded as a window for non-invasive analysis of periodontal conditions, including markers of connective tissue and bone destruction. It is called by other terms such as gingival crevicular fluid, gingival sulcular fluid, gingival crevice fluid. So this fluid contains many amounts of destructive products which are there present in it. So if there is if there is a need for any test or investigation, if there is a periodontal condition going on in the teeth, it is enough that if we take this fluid and test for the particular marker, that is particular marker for bone destruction, so then we can imagine that there is a bone destruction going on in this particular teeth. So how is it formed and how is the mechanism of this flow? So this mainly gets the gingival crevicular fluid mainly gets its component from the extracellular fluid present inside the cell. In case when there is absence of inflammation, there will be a lower vascular permeability and therefore there is low basement membrane permeability, and therefore the amount of fluid which is coming from the extracellular components present inside the cells, that is inside the gingiva, to the crevicular area is less. But when there is going to be presence of plaque, then there is going to be an osmotic gradient wherein the extracellular fluid inside the gingiva produces more, it is produced in more amount, and this increases the vascular permeability, thereby increasing the GCF production. If you are still not able to understand this, please do approach me. I will be speaking to you about this in person. All right. Next, what are the methods for collection of gingival crevicular fluid? You have your gingival washing methods, wherein two techniques as given by Skapsky and Lehner is present, or given by Oppenheim is present. Have the capillary tubes or the micropipetting methods, wherein capillary tubes of known internal diameters are inserted into the entrance of the gingival crevicular fluid, and this the crevicular fluid migrates into the tube. It is provided as an ideal as it provides an undiluted sample of the fluid which is present. You have the absorbent filter papers, and this when placed into the sulcus of the pocket, it causes little irritation, but then you get a full collection of fluid. After this fluid paper is then placed on an instrument called as periotron, which helps in calculating the amount of fluid and certain substances in the fluid as well. So the instrument is periotron. Another method is pre-weighed twisted threads. These are placed around the gingival crevice around the tooth, and the amount of fluid collected was estimated by weighing the sample thread. So coming on to the components of GCF, you have the cellular components, electrolytes, organic components, metabolic bacterial products, enzyme and enzyme inhibitors. The cellular components contains bacteria, epithelial cells, leukocytes and erythrocytes. You have the source as well as the functions, which you can go through once. Apart from this, you have electrolytes such as sodium, potassium and fluoride, organic compounds such as carbohydrate, protein and lipids, metabolic bacterial products such as aspartate aminotransferase, acid or alkaline phosphatase, and cathepsin, and enzyme or enzyme inhibitors such as, the enzyme and enzyme inhibitors are your aspartate aminotransferase, phosphatase, and cathepsin. Apart from this, you also have sometimes, if a systemic drug is taken, these drugs can also be found in the GCF fluid. Uh, so some drugs which were found by the authors were tetracycline, minocycline, and metronidazole. So this when drugs when taken systemically, we can see the presence of these drugs in the gingival crevicular fluid. So to conclude, the proper action of course, a course of action when under attack is usually to counter attack. The oral cavity is well equipped to counterattack any adverse conditions that may harm the gingiva. When the innate immunity acts primarily against any foreign invader, the specific immunity takes a more complex targeted approach to protect the gingiva. Right from its superficial epithelial layer to the innermost connective tissue, there's a line of defense that acts in harmony with the other oral structures to maintain homeostasis, to revert any imbalance that would otherwise shift the equilibrium to the disease state. Thank you.